So I'm going to just talk about some emerging therapies for myeloma. I know it's been a long day for most of you, so I'll try to keep it as simple and efficient as possible. And thankfully, when Nina presented all the immunotherapy, she basically highlighted the most important emerging therapies that are happening right now. As you all know, or as many of you know, myeloma has been with us for a very, very long time. So the oldest known case of myeloma in a skeleton was more than 4,000 years before Christ from a burial site in the Neolithic age from Vienna. So this is, so this is a disease that has been with us for a very long time. But we've had effective therapy for myeloma only since about 1960. So considering the whole duration that humanity has suffered with this disease and what we have achieved in the last 40 years, we are really making a lot of progress in the less than half a century, in fact. So this is the skeleton that I talked about. And these are the curves that I showed. So basically, myeloma has been with us for many, many centuries and eons, actually. And most of the progress has happened in the last 50 years. So you can see every 5-year period that you get diagnosed later in the course of history, your myeloma tends to do better. So already the people from 2005 to 2010 were doing better than the people who were diagnosed between 2000 to 2005. And this is just the proportion surviving. The most important part, realize that the improvement is actually happening in the first 5 years. So if you actually made it past 5 years, you get access to a lot more therapy that happens from during that time. So those are the emerging therapies that we talk about. And again, I'm sure you've seen this slide in the earlier sessions. So we have drugs that are traditional chemo, and we have drugs that now we used to call novel agents when I was a young guy. That was in the 2000s, but now they're older novel agents, I guess. And then we have newer novel agents like carfilzomib, pomalidomide, panobinostat, ixazomib or Ninlaro, Darzalex or daratumumab, elotuzumab, antibodies. And then there's a whole bunch of future drugs, including immunotherapy, and some of which were touched upon by Dr. Shah. So our goal with all these new drugs and clinical trials is to make myeloma just part of history, and that in the future we have really good options for patients who are diagnosed. So one of the most frequent questions I get is from patients: are these new drugs all chemotherapy? So the answer is I really don't know what is chemotherapy anymore. So the word chemotherapy means a chemical used for treatment. So in that case, everything that we use is a chemical that's used for treatment for myeloma. On the other hand, chemotherapy in people's minds is something that causes you to lose your hair, throw up all the time, and get a specific set of complications that are related to cellular death of a lot of collateral cells. You give the treatment with traditional chemo to kill myeloma, but you end up killing a lot of other cells, and that causes damage. From that sense, most of what we're going to discuss is not traditional chemotherapy. These are treatments that you would use just to specifically target myeloma cells. So that's the one aspect in which we've made a whole lot of progress in the last 10 to 20 years. In fact, the targeting of cells has become so much better that we are not causing so much damage to the body. But doesn't mean that these drugs don't have side effects. They have very unique side effects, just as Dr. Shah talked about the CRS syndrome and the neurotoxicity, etc. So with that, again, just as a caveat, many doctors, nurses, many people in the hospital that you go, they'll say, are you on chemotherapy? They mean, whatever drug. They're not specifically meaning traditional chemotherapy. So when we give, for example, a very targeted drug like Darzalex, which is a monoclonal antibody, as we show you in this slide, that Darzalex is we may call it chemotherapy. Oh, you need to go to the chemo room to get Darzalex. But that actually is not traditional chemotherapy. It is a highly targeted drug, and it basically attaches to this surface protein on the surface of myeloma cells. So myeloma cells have many surface proteins on the cell wall or the cell membrane, and Darzalex just homes in on one particular protein called CD38, and this is a CD38 antibody. So it is a form of passive immunotherapy in that it is a immune molecule that goes and attaches to the myeloma cell. However, it only works while you give the agent. So in fact, the main difference between passive immunotherapies like Darzalex and active immunotherapies like what Dr. Shah mentioned, the CAR T cells, is that the CAR T cells is living cell therapy, and it kind of creates an immune memory, which if we are very successful in inducing, can function like a permanent immune memory. And that's why there was so much hope about cure with CAR T cells, although it is not yet borne out so far. We are in the early stages of CAR T cells, but monoclonal antibodies like Darzalex are passive immunotherapy. Now, right now, we have one of the newest drugs that got approved for myeloma, literally within the last 2 weeks, has been something called Darzalex Faspro. And if anybody in this audience has had Darzalex, they know that the major problem is that you have to be in the hospital for a long time to get it. Especially the first few times, it's almost a whole day of getting it so that you don't get a reaction to that drug. Now we have a new agent called the Darzalex Faspro, which is a shot. It's a sub-Q injection. So it's not an IV anymore. You don't need a line. You don't need an IV infusion. And this is another thing that makes patient convenience one of the top factors, improves quality of life. And increasingly, we have clinical trials where we are using this drug to in long-term maintenance. So, when we talk about emerging therapies, it's not just new drugs that we're talking about. We're also talking about using older drugs in better ways. So, for example, Darzalex was initially approved, like all drugs are in myeloma, for people who had relapsed after multiple therapies. So people who really had no other options, those are the people who go on the initial trials. And then the drug, when initially gets approved, it is usually approved in that population. We call that relapsed refractory myeloma. It means you have myeloma that has come back multiple times, and it means myeloma that is refractory or doesn't work. Most drugs that we have don't work against that myeloma. So then you end up getting a new drug such as Darzalex, and this Darzalex started in that area of myeloma and slowly has made its way to frontline treatment. Now we have several regimens where Darzalex used upfront. So what we find in myeloma is that when we get a new class of drugs, such as this antibody, we find that when we move it in combination with other drugs that are traditionally already established and move it to the frontline patients, we get even better response. And all our philosophies is based on combination treatments and using the best treatment up front so that patients get the longest possible remission up front. So with all of this, we're going to talk about bispecific antibodies, which Dr. Shah already talked about. So I'll just skip by and just highlight what you need to know in terms of clinical trials. Then we talk about some novel targets and some new takes on old drugs. So, some of the traditional chemotherapies, some of the traditional molecules like thalidomide analogs are all coming back in a new, better improved versions. So relapsed refractory disease, we already talked about what that means. One of the newest approved classes of drugs is this new drug called Selinexor, which was approved less than a year ago. And this drug basically works best for a type of for patients who have multiply relapsed myeloma. And the way in which it works is by blocking export of proteins out of the nucleus. So essentially, when you have basically a type of myeloma called 17p deleted myeloma, 17p makes a protein. And if you don't have the 17p chromosomes or the gene in your myeloma cells, you don't make that protein. And if you don't make that protein, the cells find it very difficult to die off. So for many people, it's not an absolute lack of making the protein. It's just a deficiency in that amount of protein that you make. And what this drug does, it traps the 17p generated protein called the p53 protein inside the nucleus. So the cell nucleus tries to export it out, but this drug blocks the export of that protein, and then it stays in the nucleus. And when you give other drugs, this tumor suppressor protein is able to kill off the myeloma cell. So this is a class of drug known as nuclear export inhibitors. And most of you would not have received this drug. However, it's an oral drug, and it has certainly extended the life of many patients already. And again, like every other drug that's been discovered or invented in myeloma, it has started off with patients who have absolutely refractory disease where nothing else is working. and we hope to move it to earlier lines of therapy. And we also hope to get second generation, third generation nuclear export inhibitors and use them in a more safe fashion. So this is a study that led to its approval. It's called Expovio, that's the name. Essentially for patients who had extremely refractory disease, this drug was effective in producing a response in at least 26% of those patients. In fact, even in patients who had relapsed after CAR-T, the drug was useful. And these are patients who had a response, and you can see that their probability of survival was significantly better, and most, more than half of them were going out beyond about 18 months or so. So this is just another tool in the box, in the toolkit. And as anyone knows, as we get more weapons or more tools in our box to treat myeloma, we actually get more ways of combining those tools, and we also get more ways of more regimens built in. So for example, this drug was approved in combination with dexamethasone. Already we have combinations such as carfilzomib and selinexor, with daratumumab or Darzalex and selinexor. So when we were starting with Darzalex, we did not know that there would be a selinexor a few years later. Now that we have a selinexor, we try to see what happens when we combine both. And in patients who are Darzalex naive or who had never seen Darzalex, the response rate with that combination is 77%. For Darzalex alone, the response rate is not that high. So it means that the combination is better, and new combinations are being explored all the time. So there's another drug, again, which is not yet approved but for myeloma, but it is already available for a couple of other indications, such as several types of leukemia. And this drug is called venetoclax. This drug basically is, again, a agent that promotes cell death, and that's because cancer cells try to create molecules in themselves which prevent cell death. So the cell death in the context of what we're talking about is called apoptosis, and cancer cells create pro-apoptotic proteins or anti-apoptotic proteins. So they deplete those and they try to evade cell death. So by using agents that promote cell death, such as venetoclax, we actually can cause more cancer cells to die off. Again, in myeloma, it seems to be particularly effective for a type of myeloma known as translocation 11-14 myeloma, which correlates with a higher ratio of production of this molecule, BCL2 molecule. So, here is the efficacy data from venetoclax. As you can see, patients with the 11-14 subtype of myeloma are in this green curve, and they are the people who maximally benefit from this drug. When you look at all patients and non-11-14 patients, they did not have as much of benefit as the 11-14 patients. So this drug is particularly being explored in 11-14 myeloma patients, and we now have a biomarker for response for this drug. So that's the 11-14 marker. And if you have this type of myeloma, the 11-14 myeloma, you really need to go on a clinical trial at some point that uses this drug. It can also be obtained outside of clinical trials, but we have insurance issues getting it because this drug is not yet approved for myeloma itself. As you can see, in the 11-14 patients, the response rate is up to 40%. So one of the caveats is that there was a large trial that was being done for this venetoclax agent, and this drug, in this trial, we had to stop it early because there was significant higher toxicity in patients who got the venetoclax combination. And that's when we figured out that we really should not be using it in every patient at this time. We should be using it specifically in 11-14 patients. So the benefit seems to be limited to those patients because it is a toxic drug to use when you use it for all the patients. So it's all about risk versus benefit. So the risk of toxicity is higher in the patients who don't have the 11-14 mutation. Again, everyone here knows about thalidomide and lenalidomide or Revlimid and Pomalyst or pomalidomide. Those are all a class of drugs that begins in a family called IMiD family. So immunomodulatory family is the expansion of that. So in that family of drugs, we have another agent coming down the pike now that's called iberdomide. And the iberdomide trial was reported about a year ago at ASCO. And as you can see, patients with refractory myeloma, relapsed refractory myeloma, who had multiple prior therapies and who had prior usage of lenalidomide or Revlimid, pomalidomide, and proteasome inhibitors such as Velcade, and disease progressing within 60 days of their last therapy. So that means they had fairly bad myeloma. They had seen at least 3 of the major agents in myeloma, and they had actually disease that was taking off within the 60 days of their last therapy. They went on the study, and iberdomide was used in multiple combinations by itself, with dexamethasone alone, with Darzalex and dexamethasone, with Velcade and dexamethasone, and with carfilzomib and dexamethasone. So with all of these agent combinations, it was shown to be highly effective with about 35% response rates. And in patients who were refractory, which means absolutely not responding to lenalidomide or pomalidomide, even there, a drug or iberdomide, which is in the same class of drugs as those agents that we mentioned, has a 35% response rates, which means that this has a unique activity. And even in patients who had seen prior Revlimid or Pomalyst, iberdomide is able to induce responses. It's very promising for patients. And in this population, patients who were daratumumab and POM refractory, which means they had that both of these drugs were not working, but iberdomide worked about 30% of the time. And again, don't be disappointed by that 30% number. That 30% is what we usually see for a new drug, except for CAR T. Most of the drugs that are newly discovered start in patients who have had multiple therapies, and 30% is actually a good number for a new drug in a very refractory population of patients. As Nina mentioned, BCMA or B cell maturation antigen is the new target. It is a target on the surface of the myeloma cells, and we have several approaches targeting that. So she already mentioned about the CAR T cells, so we won't talk more about that. There are many agents in clinical trials right now. You saw the most recent update of the data which is going to be presented at ASCO. That's what Dr. Shah showed. And we talked about the special side effects. Again, at this time, I would emphasize that no CAR T cells have been approved for myeloma. So CAR T cells, to obtain CAR T cells, you need to join a clinical trial right now. Number 2, if you've already seen a type of CAR T cells, there are very few CAR T trials that will take a patient who's already seen one CAR T for a second CAR T trial. Number 3, if you have newly diagnosed myeloma, for example, or myeloma that has only relapsed one time, there are very few CAR T cells that will take trials that will take you. And most of the CAR T trials, the vast majority of CAR T trials right now are for patients who have multiply relapsed myeloma, or very high risk myeloma, or myeloma that has relapsed within a year of treatment, or something that bodes poorly for the patient. Those are the patients who need to go to CAR T. So if you're doing really well on maintenance and nothing's happening, you have stable remission going on, you cannot join a CAR T trial. So I get this question all the time in clinic. That's why just because there is something out there, we don't need to use it. In fact, if I were a patient, I wouldn't want to use CAR T unless I absolutely needed it. So if we can afford the luxury of letting the technology mature a bit, it'll be better. So the people who need CAR T right now are the people who are actively relapsing, or people who have some high risk features, or people who are running out of options. We already talked about this with Dr. Shah. So the one CAR T that's going on is the BB2121, which we think is heading towards approval. The other one that's heading towards approval is the LCAR B38, which is the other most active CAR T cell. And so a quick stop to see, I told you that we should let the technology mature, and if we can afford the luxury of waiting for that, we actually will end up using it more effectively. It is a little disappointing to many of us that CAR Ts are not giving us the cure that we expected. In fact, the other 2 diseases where it's approved, lymphoma and leukemia, we have at least a proportion of patients getting cured with CAR T. So how can that happen in myeloma? There are so many combinations that are being explored, and that was a question in the chat box too. Are people exploring combinations to prevent relapse after CAR T? Definitely yes. Multiple targets are being addressed in that. Sometimes the same BCMA target that the CAR T used, sometimes something else. Using it in earlier disease setting. Actually, instead of waiting for the myeloma to be multiply relapsed, we have to do it in clinical trials and use it earlier and see if we can do it right after transplant, instead of a transplant, before a transplant. Many scenarios where we can use it. Then change the CAR T composition. The CAR T is a living cell therapy, and there are different types of T cells that become CAR T cells. You can change the function of the cells, the proportion of cells that are memory T cells, etc. And then change the targets that you are using for CAR T, such as, we're only doing BCMA right now, change to something else, change the construct of the CAR T, change the manufacturing of the CAR T. There's so many other things that will happen. So this technology is in its earliest phase, and there's so much activity going on. And we also have to think about off-the-shelf CAR Ts that may be coming too, where you don't need to wait for the CAR T to come. And we just talked about this, ways of reducing the relapse. This was also a question in the chat box, and I said it will be addressed live. So there are many ways that patients can relapse after CAR T. So one of them is that the CAR T antigen can disappear from the surface of the myeloma cell. The second is that the CAR T itself can disappear. There's no memory of the CAR Ts, and they're gone after a while. They may not expand when they're put in the patient. There are some changes, or there could be changes in the environment of where the myeloma cell is living, which inhibits the T cells from killing the myeloma cells. All of these are mechanisms that have been identified, and these are all the mechanisms that people are working on to reduce relapse after CAR T. Again, this is Dr. Shah addressed this drug, which is called belantamab. It used to be called GSK 2857916. This is a drug that actually is an off-the-shelf option for patients with multiple myeloma, again targeting the same thing, the BCMA target that the current CAR Ts are addressing. In the initial study called the DREAM-1 study, we had a response rate of almost 60%. In the DREAM-2 study, which was a follow-on study, which actually took on patients who were even more advanced disease, the response rate dropped. But still, it is a new class of drugs. It is an off-the-shelf agent. Anybody needing it can take it off the shelf and use it. Again, right now, it is not yet approved. I'm hoping very much for my patients that will get approved very soon. So this is the part 2 study that we talked about in patients who had prior daratumumab. The response rates are actually a little bit lower, and the patients overall just a little higher. Again, I'm not going to go into details because Dr. Shah already talked about it. And then, we're talking about emerging therapies. So one of the most active emerging therapy field that we need to think about is what are the monoclonal antibodies that we have. So monoclonal antibodies are these guided missiles that go and home in on a single target, or sometimes a dual target within the myeloma cell or outside of the myeloma cell. So we have a whole bunch of drugs. Everybody has heard about this new drug called isatuximab, which is a new anti-CD38 antibody in the same vein as daratumumab, and it's got and just got approved by the FDA. So it's another option for myeloma patients. We actually don't know if patients who have failed daratumumab will respond to the isatuximab or vice versa. We have a whole lot of other monoclonal antibodies that are bound to the bound with a warhead that delivers a tiny dose of chemotherapy that's the warhead, which on the surface of the myeloma cell. So even if the antibody itself doesn't kill the myeloma cell, the warhead will kill the myeloma cell. And Dr. Shah already talked about the bispecific antibodies, and this is a whole new approach. Again, it brings an immune cell in combination with a myeloma cell. So by one end of this Velcro, it's a double-edged Velcro, in fact. So one end sticks to the myeloma cell, the other end catches the passing by immune cells, and the immune cell is now forced to interact and kill off the myeloma cells. One of the problems that happens when you have myeloma is that myeloma cells have found a way of evading your immune system. They have basically found out some way to turn off your immune system. And if your immune system can wake up, that might eventually be the way to cure myeloma. So unless we have a persistent police force in our bodies that's policing the myeloma and killing off the cells as they try to rise up, we will not be able to control myeloma. Otherwise, it's a lifelong chemotherapy. So it's better to have an immune approach, and that's the best thing that we can do. So Dr. Shah already talked about one of the CC93269, which is one of them. There are so many others in development. In fact, there are at least 7 in development that I'm aware of, and probably a few more that I'm not aware of. There is another axis on the myeloma cells. We are talking only about the BCMA at this point. There is a ligand, or there is a similar agent to BCMA called APRIL and TACI. These are all similar to BCMA in the same superfamily, and they're all stimulation of, engaging with these molecules causes the myeloma cell to survive and proliferate. So by blocking them, we can actually create drugs that can kill myeloma cells outside of BCMA targeting. So there's anti-APRIL antibodies that are in clinical trials and several other methods of blocking this. So in summary, we have a whole variety of immune strategies that are in development. Many of them have been touched upon by Dr. Shah. And again, going back, how do we put this all together? What is the emerging field? So one of the approaches is to do master protocols, where we basically take patients and profile them for minute alterations in their genes, and find out what drives the myeloma in that particular patient. It's basically a personalized approach. So you can actually find patients who are, for example, have the 1114 mutation, and we already talked about it. We have a venetoclax, that's a drug also working in this myeloma. And then at the other extreme, we may have patients who have absolutely no detectable mutations that we find. For them, we have a backbone regimen, and then we build one new agent at a time on top of that, and we test a bunch of patients with that regimen, and if we find it promising, we'll take it to further development. If we find that patients have not responded, the backbone regimen would still have conferred a response on the patient, and then we would drop that agent, whatever agent we added, and go on to test a new agent. Similar thing for various mutations that we find. So this is a interesting approach called a master approach, which actually speeds up drug development. And everybody knows now that we are speeding up development. And I think with the COVID crisis, healthcare has again become a major focus in this country, and there's a lot more money for research. Hopefully, I would think after this all ends, and there is an urgency that will help everyone with healthcare problems. So there are many myeloma precision medicine trials, and some of them I have summarized here. All of them are specifically looking at targeting specific mutations or patient-specific alterations in myeloma patients, so that it becomes a personalized therapy for a small subgroup of myeloma patients. Some of the other emerging drugs that I want to talk about is one is melflufen. Anyone who's had a transplant for myeloma has heard of the drug melphalan. That's been a drug that's been around since 1950s onwards. It's a very old chemotherapy drug, and it turns out that you can actually package that drug in a different way so that you need far less of that drug, and it's more specific to the myeloma cells if you combine it with a specific protein. And that is this new drug called melflufen. It is actually in advanced development. We have had several trials with this drug, and it is actually, again, I think fairly close to approval, which might happen in the next year or so. Another agent is the CLR-131 that Dr. Callender and others have worked on, which is again a radio targeting of myeloma cells with a specifically targeted approach. There is another drug that I'm very interested in. It's called the CD47 Don't Eat Me molecule. So when you have a cancer cell and an immune cell, sometimes the cancer cell shows a protein on its surface like this on the cartoon here, which basically tells the immune cells to stay away. So it's called the Don't Eat Me molecule, or CD47. And there are a whole variety of drugs that are being developed that actually can break down that interaction. So basically, it can either cover up this Don't Eat Me molecule so that the immune cells will eat them, or it can block that. We call those drugs checkpoint inhibitors, and that's one of the drugs that I'm involved with is called AO176. And then there is, we talked about the bispecific antibodies. We have also have trispecifics, and then we have CAR T cells, which only look at molecules on the surface of the myeloma cells. If you want to look at a target within the myeloma cell, inside of the myeloma cell, there's a different kind of cellular therapy called TCR cells. So there's a whole lot of approaches that are coming. And again, even when, my main message to anyone on this call who's got the disease to deal with is that even when things are looking bad, there may be hope from clinical trials. So always be on the lookout for clinical trials. There are many websites that summarize all these clinical trials. Again, the definitive website is clinicaltrials.gov, which is, our government-sponsored website where anybody doing a clinical trial in the United States and possibly around the world has to register their trial. So if you can actually look up for myeloma, you might find, and the places where the trial is running is already there, and you can actually contact, or you can ask your doctor to find a clinical trial for you. So please don't give up hope without thinking of clinical trials. Again, I'm sure that Dr. Callender and others talked in the morning about Profiler and other trials that look at risk assessment in a new way by beyond the traditional approaches such as FISH. And we have new tools that have been developed in the modern era. One of the things that everyone is excited about is called single cell RNA sequencing, which basically tells us what specific proteins are getting turned on within the myeloma cells. And you can actually identify patients who are either responding to treatment or failing treatment, and then look at these and then decide what drives those kind of changes. So we can actually predict and define who's responding, who's likely to respond, and who's going to fail. And these are things that will be useful later on in the next couple of years. These kind of technologies will become more widespread, and patients are going to significantly benefit. So in conclusion, I would say that significant progress has been made in the myeloma armamentarium. Even within the past 12 to 18 months, we've had selinexor, we've had the fast-acting, the subcutaneous Darzalex, we've had isatuximab. So 3 agents have been approved, again, literally within the last less than a year. We have a lot of newer therapies that are coming, immune therapies that Dr. Shah touched upon, and the non-immune therapies that we talked about, and therapies of personalizing myeloma care. We already have translocation 11-14 myeloma that might benefit from venetoclax. And then our goal is to create these and move it forward so that we use the most appropriate treatment for the most appropriate type of myeloma that you may have. It is not one disease. It is multiple different types of diseases with differing responses based on what we use. And eventually, I have no doubt that there will be a potential cure among one of these. So the main message from me is to look for a clinical trial whenever possible. It's very difficult to give an emerging medicine talk that's applicable to everyone across the board. But the persons who benefit most from clinical trials are people who have had multiple relapses. But even if you have not had multiple relapses, even if you're a newly diagnosed patient with myeloma, I truly believe that joining a clinical trial is always the best option. So looking for clinical trials, we already talked about, and then always go to a trusted website. Don't just Google. So in summary, I again thank the organizers, Jenny and Greg, and all my co-panelists for this. And again, we'll take questions now.